Thanks, Peter, and I'd like to thank the organisers, DAFWA, Ian and the GRDC for their commitment and for the opportunity to give a talk to you. So my kids ask me, Daddy, what does a breeder do? And I tell my kids that breeding is like working a jigsaw puzzle. It's about dealing with complexity and particularly identifying and targeting genetic diversity with an aim at improving performance. So how good have we been going as a nation and globally in terms of our breeding activities, given this massive complexity that resides? Well, if you look at wheat here globally, we're tracking around 1% per year per annum, which is doing quite well for a rain-fed crop compared to other irrigated and rain-fed crops globally. And half of this yield improvement is agronomic, around half of genetics. So in Australia, we're targeting around 0.5% per year genetic gain, which is consistent with some of the better efforts globally in terms of genetic improvement for wheat yield. Breeders have this capacity while selecting for very many traits controlled by many thousands of genes. And there are two largely complementary approaches that are used to facilitate improved performance and adaptation. One's around trait-based breeding, focusing around disease adaptation, water use efficiency, and the other is a more quantitative or numbers game type activity where selection is on improved yield and based on good genetics and good knowledge of pedigrees. In Australia, wheat is largely the domain of 6 private breeding companies, and these engage quite heavily with shareholders, international companies, seed companies, where we've been quite fortunate in that these breeding companies, Australian breeding companies, can leverage some of the technology and some of the wonderful science that's being developed overseas in other crops such as maize. So in understanding the drivers of yield, I guess in terms of water-limited environments, probably the number one driver of adaptation is flowering time, matching phenology for the availability and timing of water. And some lovely work, DAFWA, and published by Richard Richards, just shows quite nicely the movement away from the very early winter wheats from Europe, bringing in Indian and South African germplasm to try and bring flowering time back, try and match the finish and the availability of water a little bit better. So this has been a major driver of yield performance, and so is height as well. So where do we go now if we're sort of at the point where we've fixed the right combinations of flowering time alleles? Where is the opportunity for improved performance around traits? And this figure here summarises quite nicely, stylistically, yield for a range of environments in South Australia. This is French and Schultz, plotted against water use. And what you can see in plotting all these yields is that there's a frontier. And this frontier represents the upper water-limited yield potential. And the slope is a measure of the water use efficiency. Number of growers fall well below this line, suggesting that they're falling well below the water-limited yield potential. And can we use genetics and agronomy to try and lift these growers, try and move up beyond our 1% genetic gain? And so what we'd like to do is, using good knowledge of physiology and genetics, develop varieties which give us the capacity to move beyond the current frontier, around 22 kilograms per hectare, up to 24, 25 kilograms per hectare. So can we do that? And can we do that around existing agricultural systems? And so there's a genetic component which is critical in all this, but we can't dismiss the importance of agronomy. And so in building and lifting these growers in many of these environments up to their water-limited yield potential, capacity to reduce soil-borne diseases, to improve water uptake. And so the benefits around crop rotation, using the capacity for canola and other crops to reduce the build-up of root diseases. Genetics, the capacity to improve water uptake through improved salt tolerance, particularly in crops like durum wheat. And as we heard from the previous speaker, the capacity to improve root growth in acid and low pH soils. So we have all this genetic diversity, we have this target around water-limited yield potential. You know, we're almost drowning in our gene pool, so to speak, with the diversity that we have. And so the question becomes, well, what traits should we be targeting? And I guess the group I work with in Canberra has modelled their activities around yield potential in woodland environments around a framework a framework focusing on water use efficiency and improved water use. And so I'm going to give an example of some of our work where we're trying to introduce new novel diversity not present within current breeding programs, to try and lift our capacity, in this case to maximise water use. And the importance of water use is probably best demonstrated agronomically from some work from David Hall 10 years ago, where for the same variety using 2 different nutrient regimes, David was able to double the yield of a wheat crop by trying to mimic or modify the early growth, the early vigour of the crop. The data is quite lovely in the sense that the 2 treatments had exactly the same water use, around 360 mils, this is in Esperance, but almost half the water that fell on the low vigour treatment was evaporated away. So there was less water for evaporation, in contrast to the more vigorous crop, where there was more water conserved, less water evaporated. This meant more water for transpiration, in turn, greater yields. So that's a nice example around an agronomic innovation to try and improve or reduce water loss through soil evaporation. We've been working at CSIRO to try and do this genetically in looking at diversity that's out there. And our first efforts were around some fairly novel but not particularly vigorous germplasm we accessed from India and China. And Tina Botwright, working with this material, this is back cross 2 material, at 2 sites showed for sister lines, quite different in vigour, a large increase at Wongan Hills and Merredin in biomass, which translated to increase in grain yield. So we were quite excited. Given the quality of this vigour germplasm, not being particularly vigorous, there was some capacity in moving genetically to improving water use efficiency through greater vigour. As part of this activity, we identified other sources of germplasm: India, Israel, Argentina, material that was quite unique in breeding programs overseas and, from what we understood, unrelated through pedigrees. So these were likely to be vigorous lines that had different genes for vigour. And so we undertook a maize-type breeding activity to try and bring together all these different sources of vigour from all over the world using a technique called recurrent selection. It's a maize-like breeding strategy. And we've moved from an example here of cultivar Annuello to our first cycle of selection for vigour. We're now to cycle 6, but you can see already at cycle 4, this is for wheats for exactly the same grain size, we're seeing a 50% increase in leaf area just through genetics. This material is very vigorous, has large leaves. We've also identified that this material, because of its large leaf area, also has a very large root system. And this large root system is characterised by a large amount of fine hairs. So fine root hairs contributing to increased root biomass and the capacity to better explore the soil surface. Here are some of the lines for showing root biomass in tubes, cycle 4 vigours compared to beecher barley, and some of our better Australian wheat varieties. This material, when tested under nutrient limiting conditions, performs very, very well. Here's some work from Richard James showing that under phosphorus limited conditions, the high vigour material has this capacity to maintain early growth. And from Iro Poulter, capacity also for maintaining increased nitrogen uptake with selection for early vigour. Bob French has had some of this material at Merredin as part of his MEF activities, and is also showing the capacity for this material to recover after a false break. So a very dry start to the season. We're not sure what might be driving some of this capacity to recover, but certainly there's some interesting capacity in this germplasm that may be of real benefit through selection for early vigour for West Australian soils. And then work that we're just about to commence with the GRDC, where we're looking at the capacity for greater shoot vigour and potentially greater competitiveness below ground to completely suppress or at least reduce the impact of weeds in our farming systems. So we have really good evidence from work with Gurjit Gill at University of Adelaide, Peter Newman, DAFWA, and more recently Mike Faulkner in South Australia, showing that some of these very vigorous wheats completely suppress weeds when sown with rye or in model weeds such as oats. So we have this capacity around understanding which traits are likely to be important. We have some understanding of where the genetics may reside. Our weakness is phenotyping. So these are complex traits which interact with the environment, and the GRDC have been quite instrumental in developing this capacity for high throughput phenotyping under targeted stresses. And the first of these is the managed environment facilities, co-located across 3 sites throughout the Australian wheat belt. These sites are at Merredin, Yanco and Narrabri, and reflect 3 very contrasting soil water availabilities. So very different historic rainfall patterns. And by using these, or I guess taking account of these quite uniquely different sites and regions, we have the capacity to check or test for trait value and also assess the capacity for high throughput phenotyping for particular traits in particular genetic backgrounds across these 3 sites. Coinciding with the establishment of these 3 managed environment facilities is this capacity for high throughput phenotyping. And Lynn Jones may have talked about some of this yesterday. This amazing technology which is being developed and close to delivery, which allows us to screen large numbers of lines, large nurseries, very quickly for characteristics such as leaf area. This is using the LiDAR system, the light radar system, where in the space of 3 seconds, we, for a given plot, can identify the number of heads and potentially also the amount of fractional light intercepted. Deploying capacity for Everest temperature thermometers out of planes, out of balloons, and out of helicopters has also given us the capacity to really assess critically and reliably water use in these managed environment facilities. And so if you understand water's driving most of our performance, capacity to get at water and use that water more efficiently, transpiration is going to be a critical driver of the quality of our germplasm and our ability to perform. And probably the best signal of transpiration is Canopy temperature. And so here's a simple biophysical process which can be assessed very cheaply and rapidly over large areas, up to 3 hectares at any one time. And from that we can very closely look at, for a trial of around 1.5 hectares, differences in Canopy temperature across all lines treated and irrigated the same way. This is a mapping population growing at Yanco under controlled irrigation. And we've improved the heritability, the confidence we have in understanding the underlying genetics. We've improved that capacity from around 10%, so 90% noise, 10% using traditional methods of measuring Canopy temperature, to around 80% using these helicopters and using planes to get one-shot images of Canopy temperature. So real powering going forward around the use of some of these new technologies when undertaken under very controlled, specific irrigation regimes in the managed environment facilities. And this is just to highlight from some of this work, just the relationship of Canopy temperature against a measure of water use efficiency. And it just shows, even for a very small range in Canopy temperature of less than 1 degree, we have incredible confidence in being able to determine differences genetically here at the pedigree level in transpiration efficiency across a range of representative Australian wheat varieties. So real capacity for high throughput phenotyping and early generation culling in commercial breeding. So moving to other tools which hopefully will move us towards this 25% yield gain. And I guess it's no surprise that biotechnology is a major driver of our success of recent years and will continue to be so. Wheat is a quite an interesting species. It contains 3 genomes. The genomic size of wheat is 18 times that of humans. And of that wheat, so much of the DNA in wheat is redundant DNA or junk DNA. And so for a breeder, using wheat and looking for those winners is a bit like looking for a needle in a haystack. And so how can we help our breeders? How can we help our growers identifying what will be the next mace or the next wild catch? And so these tools, molecular genotyping tools, are becoming commonplace in the breeding programs. They are the main, some of the main engine drivers of selection for many of our important traits. And what they've allowed us to do is allowed us to target the genetics that drive trait variation removing that issue of heritability and misclassification of the phenotype with the gene. They've allowed us to do it very early in part of the breeding process, so it allows us to enrich populations, so only the better, more likely successful lines will be promoted. And they've done it in a way that the cost is coming down considerably. And a slide from Intergrain just highlighting how this is being deployed, these technologies are being deployed in a very relevant to West Australia breeding program, using technologies developed in the United States with Monsanto around this robotic seed chipper, where there's capacity to access and assess DNA from many thousands of lines in a very short period of time. So the engine is there, the capacity for screening for markers, molecular markers cheaply and reliably is there, but there's also the requirement to understand which of the genes are underpinning the important traits we're targeting. Late maturity alpha amylase, disease resistance, grain yield. Some of these traits are quite complex and so controlled by many genes, and so we've undertaken this capacity to develop a very large integrated cross, magic cross, which includes varieties from around up to 16 varieties across the world, and using the powers of genetics, looked at recombination across this material, and from up to 2,000 lines assessed for various traits, we've got a very good understanding of what the genetic architecture of some of our more important and genetically complex traits are like. And so the example here is around coleoptile length. Just shows you, just highlights various regions across the genome associated with something as simple as shoot length below the soil surface, establishment, up to a dozen QTL. And what it highlights is that across our varieties, which don't differ remarkably for coleoptile length, they contain positive alleles of different regions. And so you've got YIP-P with this 1AS QTL of large effect, and you've got Westonia with this 4A QTL of large effect, Baxter 3AS QTL of large effect. So a number of QTL, a number of alleles that we didn't understand or know previously are there, and there's now capacity to identify those and use current genetics to identify and bring these together as part of our enrichment program, selection program. And certainly enough, as part of an existing project, ACR project, we've been looking at some marker assistant enrichment, and we're identifying germplasm with coleoptiles of around 4 inches in length. So capacity for very deep sowing. These are semi-dwarf wheats. And of course, it's nice having long coleoptiles, but really it's not particularly useful unless we see it used appropriately in the appropriate target agronomic system. And so some work with James Hunt, where he simulated for a site in Victoria, mean yield about 1.6 per hectare, but with good agronomy, with good water-saving agronomy, we saw, with no-till, an increase in yield, fallow weed control, another increase in yield, pea crop. But with this capacity to sow early, which we struggle with our current varieties, and with a long coleoptile, we have this capacity to make better use of this water, make better use of this limitation to improve water use efficiency and increase yield. So coleoptile length in the right farming system, this is southern New South Wales, but it's relevant everywhere, can give us the capacity to further increase our genetic and our national wheat gains. So moving towards the end of the talk, I think, how much? 5 minutes. Lovely slide from Don Duvik, which just highlights the yield progress with maize in the US, and just how increasing yield has coincided nicely with the development of hybrids. And it's an area that that's a messy slide. It's an area where I see incredible potential in Australian wheat. The development of hybrid wheats to rapidly integrate new genetic diversity, but also develop high yielding varieties through heterosis. And also to introduce diversity quickly. Issues around the development of heterotic gene pools and the cost of seed production are going to slow delivery, but we're moving along in a very, very positive way here. So the last area of the talk is around GM. And I guess I just wanted to highlight here that we currently make good use of diversity within our crop species, our cultigens. And this is diversity is developed through domestication, hybridisation, selection, and in some cases we use induced mutation. Some novel techniques have since developed in specific hybridisation and somatic fusion, but in moving towards other organisms, we have this unlimited potential to tap into other diversity. Not only that, new genes, but also precisely target expression for these genes. And this slide, this is from 2011, just showing how over the past 15 years there has been a linear increase in the adoption of GM crops globally, both in developed and developing countries. And in the case of cotton, soybean, maize and canola, quite large efforts in the breeding activity have been tailored to movement away from conventional to the use of biotech and particularly GM tools for introducing important alleles for performance. So in wheat, we have the capacity with GM to develop single insertion events. We can silence genes. We can insert multiple genes, target biochemical pathways. We can target gene-specific locations without disrupting an already adapted genetic background. And we have the capacity to also introduce gene cassettes or mini chromosomes containing many targeted genes, say for example, many important disease genes. So in finishing up, the step changes through gene technologies I see in wheat around GM are really around bringing hybrid vigour to non-hybrid crops like wheat, the capacity to develop pure breeding hybrids through apomixis. It's a common technique in wild species. Capacity to improve photosynthesis using technologies, photorespiration capacity, as we see in maize and other C4 crops, water and nutrient use efficiency, and the capacity around improving stress tolerance. An example here is around aluminium tolerant barley. And my vision as to how this will be deployed around existing breeding programs is demonstrated here, where from 1990, biotechnology delivered the first link markers, the capacity to identify and clone major genes. We're moving into QTL pyramiding and genomic selection, tools developed and adopted from the animal breeders. The first GM crops, wheat crops, will probably coincide around 2028, 2030, and they'll focus on resistant gene cassettes, herbicide, nutrient use efficiency, simply inherited traits, major genes of large effect, but ultimately moving into some of these more complex traits like C4 photosynthesis, the potential for frost tolerance. And while these are all goodies and while they're going to improve water use efficiency, improve genetic progress massively, they cannot be done without the ongoing selection, the bread and butter of our breeding programs, the targeting of multiple genes efficiently and in a directed way. So breeders are going to be here for a long time yet. So in summary, just wanted to show you my favourite slide first. This is how genetics works. And just to remind everybody that it's critical in maintaining genetic gain that we maintain and understand our genetic diversity in a way that can be targeted around what it is that's limiting productivity, particularly in rain-fed systems. Quality, robust phenotyping is the limiting factor, I think, and we are developing tools to help breeders to better understand the underlying genotype or phenotype to get at the underlying genes, underlying genotype. None of this is valuable unless we can think about the adoption and delivery of varieties in the context of the agronomic farming system. And so having that synergy around agronomic systems and systems knowledge is absolutely critical in maintaining our capacity for continued genetic gain. Biotechnology is going to become the workhorse, I think. I think the cost of markers coming down, we're close to the capacity for sequencing the wheat genome. We're going to be in a really good space in 10 years' time to really get at those genes that underpin the important traits that we're targeting and perhaps identifying new alleles that aren't present in our existing breeding programs through this knowledge. And finally, I believe GM winter wheats are coming, but I think are looking at 2025 to 2030, and they will largely focus on high return traits initially. So with that, I'd like to thank the GRDC for their support, and I'd like to thank Ian, DAFWA and the update organisers for their invitation to come and talk. And finally to my colleagues and friends and those of you who contributed slides and ideas. Thank you.
Thanks, Greg. Kadambod Siddiq from University of Western Australia. I have two brief questions. One is, you mentioned 0.5% rate of yield increase throughout Australia. But if you go and focus on the low rainfall areas, below 325 millimeters, I think that 0.5 becomes much lower. So the question is that, is there much room for further improvement genetically? Yes, there is scope for agronomic. The second question is, what's the role of synthetic wheat? We heard a lot about it, a lot of noise, and where is it?
Yeah, good question, Siddiq. The first question, it's interesting when you look at the literature and look at the data around the low rainfall zones, there are years when performance is better and there are years when performance is not as good. And so I guess for me it's about targeting the right diversity and targeting around what it is limiting productivity in those woodland environments. And is it access to water, is it timing of sowing? So I think there are real opportunities there in improving productivity around low yield potential environments. And I think too, this is where I agree that this is where agronomy is going to be absolutely critical in our capacity to keep what little water we have and use that water more efficiently, using that example I gave for southern New South Wales, where water is precious, and yet with the right agronomy, the right management, we can double the productivity and water use efficiency through good agronomy. The second question around synthetic wheats is an interesting one. Synthetic wheats have been around for 25 years, and so for those who don't know, synthetic wheats are essentially wheats that have been remade. Bread wheat is a 3-genome beast. It's effectively a durum and a D-genome progenitor. And so 10,000 years ago, nature got lucky and bread wheat was developed. And so with that coming together, it's believed there are probably only a handful of D-genome progenitors which were used in making bread wheat. And so we have a massive bottleneck in the allelic diversity in the D-genome. Lots of diversity in the AB genome, the durum genome, but in the D genome there's not so much. And so there's been a strong effort from Simmet and others to try and bring in diversity in the D genome through the development of these synthetics. And I guess I'm not convinced of the value of those at this point, partly because it's been very much hit and miss as to which Taoshi progenitors were used. But secondly, there's been so much rubbish that's come in selecting for favourable alleles in the D genome. Linkage drag and other factors are bringing in other negative alleles which are perhaps reducing the obvious value of this new diversity coming out of the D genome. That being said, I see considerable potential in the AB genome. So the durum wheat genome is quite spectacular in terms of yield progress. It's double that of bread wheat. Which is quite remarkable. And so there's amazing diversity around radiation use efficiency, around photosynthetic rates, those sorts of things sitting in the AB genome, which is untapped. And so I see the potential in synthetics around the capacity to bring in novel durum wheat alleles, less so than the Taoshi because of that issue around linkage drag. So a very long answer to a very simple question. Thank you.
Just one more question.
Steve.
Greg, Ken Flower, from UWA. Early on in the talk, you put a lot of emphasis on high early vigour in relation to yield as a sort of way to breed for higher yield. And obviously under controlled conditions that would hold. My question is, have you investigated the relationship between early vigour and yield under many different growing environments? Now, I ask that question because, just for example, we've got a long-term trial, and often the treatments with the highest early vigour are the lowest yielding. So, because they're using that early water, and there's often a negative correlation when you go into some of those drier environments. So, have you looked at that relationship and related it to what you're doing?
Absolutely. No, in our assessment of the value of vigour, we very much take into account potential environments a crop is likely to experience. And certainly Steve Jeffries and I have talked quite a bit about what happens under very water-limited environments where you're talking yield potentials of 1.5 tonne. And I imagine very little is going to give you a benefit in those conditions, perhaps transpiration efficiency. But the risk is you do run the risk of exhausting your water under those very low yield potential conditions. I guess for the traits we're targeting, we're talking about average seasons, 2 to 2.5 tonne, 3 tonne. And I don't see in our understanding of the literature and understanding of the data we have around the managed environment facilities, that vigour is going to be too much of a constraint in those average type seasons, particularly Mediterranean type environments, and particularly where you have some starting moisture. So I guess there's still a little to learn, and it may be that the vigorous suites we're developing are perhaps too vigorous, and we run the risk of them over-exhausting the water. But I guess time will tell as that material is made available and released to the breeding programs for assessment. But it's a good point.